Before we start the lecture, let's pay homage to Atal Bihari Vajpayee. One thing related to the science, that earlier Lal Bahadur Shastri had given Jai Jawan, Jai Kisan, and to that Atal Bihari Vajpayee added Jai Vignan. So that is the thing, and then he did many things in science, as we know. The roads all over the country connecting each other to facilitate many things, number one. Number two, Pokhran 1998, the office, and many sanctions were put by countries like America and all that, but he faced it very bravely. I know many BRC and TIFR people have suffered then, including the chairperson Chidambaram could not go to USA for a planned meeting also. But then it's all happens. So we'll stand for a minute and pay homage to Vajpayee. Thank you. I cannot avoid temptation of telling you one story about Vajpayee. It was 1986. There was Marathi Sahitya Sammelan at King George High School, Dadar. And I had helped the organization by managing one advertisement worth 25,000 rupees. So I was a special invitee of them, and I was given a seat in the third rank. And then just 5 minutes before the program began, one gentleman came and sat in the second row just in front of me. And then the secretary of the organization, Mr. Daji Bhatodekar, he came and said, "Sir, aap manch pe aaiye." He said, "No, I have come here to see how Marathi Sahitya Sammelan goes on. So I am not going to come there on the dais." Still, Bhatodekar was not happy. He went on the stage and requested. Gadgil said, "This is not in planning, so we will not bring him on the stage." Bhatodekar did not come back again. Gentleman sat for the entire program, and after the end, immediately he got up and went back. He was Atal Bihari Vajpayee. Today is the 29th lecture, and then second lady only out of 29 lecturers. It is Vidita Vaidya. Vidita is PhD in neuroscience from Yale University, New Haven City, USA. And then she joined TIFR in the year 2000. So last 18 years she is working in TIFR in various capacities. Since 2005 she is a full professor. Vidita works in the Department of Biological Sciences, and then her research group at TIFR is interested in understanding the neurocircuitry of emotion, its modulation of life experience, and mood modulatory drugs. Third, the alterations in emotional neurocircuitry that underlie anxiety and depression. I would not read the entire data, difficult to understand also, difficult for me to pronounce it also. But then through her lecture, she will try to explain it as simple as possible, because this audience is of common discipline, not specialist in your subject at all. Not that everyone is from science discipline, but they are interested in science and new subjects that we organize here month by month. So I would request Mr. Sharad Kale to welcome Vidita Vaidya. And now Dias is yours.
Thank you very much. It's a pleasure to be here, and it's an honor to be the second woman, hopefully the second of many in a line of more women to come after this. We mentioned the former Prime Minister, who was known for his incredible oratory skills and an ability to reach out to very diverse audiences. And one thing that I'm struck by as a scientist is that we don't get the chance to talk to people who will improve our oratory skills, which will really challenge us to be able to explain what we do as simply as possible and still be able to share the magic of the discoveries we get an opportunity to be part of. So hopefully I'll try to do as good a job as I can to introduce you to why I think the brain is as interesting as it is and why it's kept so many people so interested for now centuries and centuries. Okay, so I think there's a neuroscientist lying in most of the people in this room. You will be interested in why you behave the way you do. If you're not interested in your own behavior, you will certainly be interested in the behavior of your spouse or your child and wonder, what did I do to cause this reaction, or what is the reason that this has happened a particular way? And if you have children preparing for an exam, so in some way or the other, parents, human beings, we're all interested in what makes our brain tick. What I had a title which was the emotional brain, but you see that my title has changed. Okay, my title is now Neuroscience Then and Neuroscience Now. It's a meandering walk through the neuroscience gharanas. Now, there is no such thing as a neuroscience gharana. It's a coined up, made up word that I have just made up, inspired by the gharanas of Indian tradition, which is that there is a history of a Guru Shishya Parampara, and people have been asking in music and experimenting in music for a long time, much the way that people introduce you to the idea based on inspiration from the musical gharana. So if you were to introduce musical gharanas, you would say every gharana has distinct features, differs in the way musical notes are sung. You know who is the guru who set up the tradition. You know who are the shishyas of the current time, who are the masters and the exponents of that gharana. Now, how will you do this in neuroscience, right? So this is why, first and foremost, that there is no such thing as a neuroscience gharana. I'm going to attempt to create one. Okay, it's an incomplete and imperfect meandering walk. Means I'm going to go somewhere this way sometimes and I'm going to go other directions sometimes, to identify neuroscience gharanas and to look at who the original gurus or people who set up those ideas were and who are their modern day shishyas. I will also tell you that this is a biased approach because it is the history of modern neuroscience is short. It has happily forgotten the East. It has happily forgotten philosophers of the East. It is a colonial biased history with a strong representation from the West. But I don't want you walking away with thinking that neuroscience has not perplexed people across the globe. It has, and many philosophers from the Indian tradition and from the Chinese traditions have puzzled about what makes us us. What is it that is the defining consciousness as a question has been common to philosophers across the globe. It's a central, unanswered question of neuroscience till today. We still do not know the circuits in the brain that produce our sense of consciousness. So let me start with one of the oldest gharanas. Okay, this is the grave digger gharana. There is a reason I call it the grave digger gharana, because the central question that this gharana wanted to know is, what is the brain here that is actually constituting the brain? And if I have to find the master originator of this gharana, then I have to say it's Leonardo da Vinci. Okay, partly because he was courageous enough to look. Now, this was a very difficult time. You did not get access to the brain easily. This is also the time where, it's a Renaissance time, but it was still heresy to open up and dig up a grave, pull out the body, cut open the skull and then take out the brain. This was not okay. It was not considered acceptable. Now, this is interesting to think about because it was not heretical in the Indian system to do dissection. So if you look at what Sushrut Samhita, Charak Samhita has, there are dissections and there are drawings. So the idea of cutting up a body and looking at the internal parts was not heretical within certain system, but was heresy in parts of Europe at that point in time. which is why people who took out bodies and cut open, now you don't even think about it. Will you go to a doctor who has never cut a cadaver? You will think 10 times before it. You will say, I don't want to go to a neurosurgeon that has only practiced on, maybe a computer simulation, or at the maximum you want somebody who has actually had some experience. That's a natural thing, right? We don't even think about it. And yet, how many of us would be willing to leave our bodies to medical science? Because without leaving our bodies to medical science, science doesn't progress. And it took a long time. We take it for granted that people were dissecting cadavers. But the dissection of cadavers as a practice is relatively recent in the history of modern science. For the longest time, people, they managed to get something that somebody took out of a grave and then they were able to look. And which is why the drawings are not perfect. They're incomplete, they're imperfect, partly because they didn't have, look at the, you'll see, but yet it's pretty remarkable. And if you look at the brain, this is Da Vinci's skull sectioning in 1489. So that gives you an era, that period of time. There was really no place to store the brain. So once an individual dies, very soon things putrefy, they go bad. That's why you can't actually look at the brain very easily. But have drawn and understood without having access to this kind of material. And I want to use this as an example. Most of you will recognize this particular painting. And it's a remarkably beautiful painting by Michelangelo. But now I want you to look at the cloud on which God is lying. Look at the cloud on which God is lying, because I'm going to show you what the cloud looks like. Outline of the human brain in a sectioned fashion with what would have been the pituitary gland sticking out at the position at which the angel cherub's foot is sticking out. That is the location of the pituitary. So if you look at this cloud, and it's absolutely, that's the cerebellum right there. Okay, right down to almost a perfect outline. Now we can't go back and ask Michelangelo, did you mean to draw a human brain in the cloud shape on which God lies? You can't ask this question. But it begs the question that what was he thinking when he drew this? Or is this something we have just discovered years later and it's fluke? He had drawn so many human brains that he just by fluke drew it. But most clouds don't look like this. Most clouds do not have that shape. And it is actually a very interesting observation made by a graduate student, medical student, F.L. Fischberg, on which he wrote an entire paper on God is a product of the human brain and imagination. That is heresy at that point in time. Michelangelo had said that he could not have been painting in the Vatican. It would have been absolutely heretical to say that, right? But was he suggesting that? What was the suggestion behind this? Is an interesting source of debate. We will never know. But without the gravedigger gharana, we would have known nothing about the nervous system as it stands. We would not have had our first idea of what the nervous system looked like. And so we really, when you think about the nervous system, this is again from Da Vinci's drawings. And incredibly beautiful detailed drawings, late 15th century drawing of a skull. That's what it looks like. It weighs only 3 pounds, 1.5 kg. Your sense of self, self-worth, poetry, consciousness emerges from this circuit. Thank you. If you were to cut a section and look, and then zoom in, that's a little structure that is responsible for learning and memory. Right there. 100 buildings. That's an artist's rendition after a certain point. And I'll show you what real neurons look like. They're not very different looking from those cartoon drawings. But we would not have gotten where we know about how neurons look without what is called the father of modern neuroscience, which is Ramon y Cajal, Spanish pathologist. And his job was to use these microscopes, which are really simple microscopes, and draw what he saw. That's all he did. Beautiful ink drawings. And then he wrote books about what he thought the brain did. And he got a lot of it right without actually having done the experiment. So now we discover after doing the experiment that he actually got it right more than like 100 years ago. But this is what his drawings look like. He would see these cells, and this is actually a section from the cerebellum. And so in a sense, in drawing, he started figuring out that these things were connected to each other. At that point in time, he and Camillo Golgi, who's the guy who came up with the technique that allowed these neurons to be stained, all of this would not have happened without that dyes and the food additives industry. All those dyes that were used for staining textiles were also poured onto brain sections, and they happened to stain neurons. So you could start using dyes from the chemicals industry, apply it over to this field, and suddenly you can see things. Otherwise, these things just look like, mush. So when you put them underneath the microscope, you start seeing cells. That's when the debate started. Already the idea that there are cells was there. But when you look in the nervous system, they look like nothing like the cells in your skin, which are usually nice and round. These things have all kinds of stuff coming out of them, and they look like nets. So Camillo Golgi said this is just one continuous net and everything is connected to everything. So that was the network theory. And Cajal said no, they're individual cells and they talk to each other. That didn't look like it. He found it very hard to prove that they talk to each other because he didn't have electron microscopy. That didn't come till about 40 years later. So these two, both guys shared the Nobel Prize because the debate could not be solved. And anyway, without Camillo Golgi, Ramon y Cajal would not have been able to look at this stuff. Okay, so this is 1906 for their Nobel Prize, shared Nobel Prize. It's much later that we saw synapse. And we knew that, Cajal was right. These are individual cells. Some of them are incredibly large. So the cell that controls my ability to do this with my foot is going to be 1 meter long in its projection because it's sitting in my motor cortex but has to project all the way down to the bottom of my spinal cord. Now imagine, so most times when you think about a single cell, you're thinking 10 microns, tiny, 20 microns, very small. Micron is 10 to the power of -6, okay, meters. So it's small. It's a thousandth of a millimeter. So it's pretty tiny. That's the kind of cell size that you're thinking. Here now you could say that same cell has membrane and cell size that is long enough to cover a meter in the case of a human, several meters in the case of a giraffe. So this is a non-trivial problem, which is why these cells, once you make them, you don't want to get rid of them very easily. Because unlike your skin, which you are shedding right now, many of you don't know this, but you're shedding your skin. That's all there. You don't shed your neurons. If you shed your neurons like that, that's a bit of a problem. You can't replace them that easily. Partly because unlike this layer of skin, which is small cells just attached to each other and they keep replenishing, you can't replenish those neurons which have to make it across a meter. Your little baby, it has less distance to travel. Baby born doesn't start walking right away because that circuit is not laid out. It has to learn to walk. Once the walking circuit is stabilized, you don't have the cells in your body. These are your only true companions through your entire life. Everyone else whom you think is your life partner through life, hopefully will be there throughout life, but may not really be around. And they were not around when you were a baby. And most of the people who you've had in your life come and go. Hopefully you have companions that are there for long enough. The only real companion you actually have are your neurons. Because every other cell that you have has turned over. Your bone is replacing every 3 months. Your skin is replacing faster than that. Okay, your liver is replacing its cells. So everything is turning over. So if everything is turning over, what are the cells that remain with you through your entire life? It's your neurons. Okay, most of those cells are the only cells that are going to hang in there throughout. And so Cajal had these beautiful drawings he made of these cells. What do we do now 100 years later after Cajal, right? Did he solve everything? No, like I told you, we could not even show for another 40 years after he had the idea that there actually is a synapse, because to show it and prove it, you needed to see the gap. The gap is angstrom, so it's so small you can't see it without an electron microscope. So somebody will say, oh, you think there is a gap, but you show it to me. I want to physically actually see it. To physically see it, you have to wait for the electron microscope to come into picture. So you wait. So fields move, questions don't disappear sometimes. The questions are the same that existed 100 years ago. The tools of the trade dramatically change. So Cajal was sitting with a simple drawing, and he's the father of modern neuroscience because he had a great imagination. What about today? We are still painting the brain, except we are not painting it through a light microscope. We are genetically painting it. So we have now genetic tools that can say, I want this neuron to express green fluorescent protein. That one I want expressing yellow or pink fluorescent protein. So you can use genetic tools to get your neurons to light up with fluorescent dyes. And by doing that, you can render the entire brain visible. And these two guys, Joshua Sains and Jeff Lichtman, are the inheritors of this gharana. That's my picking. There are many other people in this field, but I just picked those two because they made the prettiest example of painting the brain, according to me. This mouse is called a rainbow mouse. It's called a rainbow mouse because the brain is painted in rainbow colors. It's just genetic tools. All of these fluorescent proteins are actually there in the red algae and the green algae and the fluorescent algae which are there in the water. Which if you're lucky enough to go to a beautiful seaside, sometimes you will see some glow on the sea, and that's because there are these fluorescing cyanobacteria, cyanoalgae, etc. You can take out the gene that is responsible for the fluorescent protein in those things, use what is very cool cut and paste microbiology. You can cut it out and you can stick it in. Now you have to stick it into the animal that you want, and that I have just covered about 50 years of molecular biology that allowed us to chop things out and stick them into other places, without which, by the way, insulin would not be available to most people in this room if they were diabetic. This cutting and sticking was a technology that dramatically altered how we could produce all kinds of proteins. So now we can cut and paste, and because of that, what in the past required us to have a light microscope to draw and stains from the pigment industry looks like this. There's 3 of those cells, 1 in pink, 1 in dark blue, and 2 in green, and then you can see what they look like right next to each other using genetic tools. And this is the paper that actually led to this. This is the rainbow mouse, and as pretty, this is how it looks. This is a section through the rainbow mouse's brain, and you can actually see the entire architecture of this gorgeous looking nervous system, and it looks remarkable. It's very, very beautiful. We know in the human brain there are 10 to the power of 11 neurons. Each neuron receives 10,000 synaptic contacts and makes 10,000 synaptic contacts. So that's 10 to the power of 15 synapses in the human brain. That's an estimate. Each cell is slightly different from each other, even though there are groups of cells that are similar to each other. In their electrical engineer's nightmare. Because if you want to solve this, who's connected to whom? What kind of a switch is it? Is it on? Is it an off switch? Because things can actually cause the responding cell to activate or cause the responding cell to inhibit. Don't know the network. The big brain connectome project is attempting to solve this. They want to, in the next 10 to 15 years, find out the wiring map of the human brain. Now, this will require computer scientists, people who can do complex algorithms. It's going to require people who will do the hard work of actually looking in the nervous system, and yet it would not have been possible for a while to even take on this kind of a project. It's just too challenging, too difficult. Like the Human Genome Project, which involved getting the entire genome, but you realize that one person's genome is not like the other person's genome. So just because we get the wiring diagram of one human being, it's not going to clone the wiring diagram of another human being. We know that already. And we also know that just getting the wiring diagram will not tell us everything. For example, for the worm, we have known the wiring diagram. We know there are 302 neurons. We know every neuron that is connected to everything else. The whole map is known, and yet we can't predict function fully. So even once we get there, we're not going to have function automatically. It's going to just be one piece of information, but it's a huge challenge. And the US has thrown a large amount of money at this project, and different countries have major. So there's the Blue Brain project, there's the Connectome project. So Europe has a large project. Japan has a major project. So does the US. India is deciding. I would not bank on this Connectome project, partly for the following reason, that just finding out who's connected to whom doesn't tell you how it works. If that's all it was that was required, we would have been able to tell everything about the worm, by knowing its connecting plan, and we don't yet. We can't even tell everything that the worm is able to do based on that. But some things have become very, very easy, and those things that have become easy, which will change the way we can look at the brain, has come from what is called the Connectome project from Carl Deseroth's lab.
We're heading into the center of a mouse's brain, into the hippocampus where memories are formed. Looking up, you can see neurons projecting to the surface of the brain. This is the work of Carl Deseroth and his team at Stanford University. By making the entire brain transparent, they were able to image it using a light microscope. They call Existing techniques for studying the brain's wiring are often limited to looking at very small volume text. The advantage of clarity is that you can label lots of molecules in whole brains.
I want to show you this for the following reason. You know, when you take a brain out, you have to normally cut it to be able to even see neurons in a thin section. The section thickness is usually 50 microns. Okay, microns, remember, is 1,000th of a millimeter. So really thin sections, and you're cutting. Now imagine even a mouse brain which is several millimeters. You'll cut and cut and cut and image and recreate. Multiple PhDs and multiple people's lives will go, and you still won't get the full connecting diagram because it's just that hard. And cells connect from the front to the back. They have to, because they have to control things across long distances. So to find out who is connected to whom, and if each cell is making 10,000 contacts and receiving 10,000, this becomes a nightmare. It's like a needle in a haystack issue. You're trying to find one cell and find its connected. One of the biggest problems is because you have to cut it and look at it. What if you could look at it without cutting the brain? This was not possible because it's full of fat. Your brain is full of fat, and fat occludes things. So that became a serious issue until you had this technique called clarity, in which what Carl Deseroth and his lab did, and I just want to show you this part.
So how do you make a brain transparent?
He made the brain transparent.
The thing that obscures the view is fat. Lipid layers surround each cell. To remove them without disrupting the cell structure, the team used a hydrogel to create a mesh to hold the rest of the components in place. Then they could clear away the fat. This is a mouse brain before and after.
There's a brain on the right-hand side. You see
the brain is now transparent to light, but it's also permeable to molecules, which means scientists can add molecular markers to highlight specific features.
Because it's transparent, you can see straight through it.
In this 1 millimeter block of hippocampus, excitatory neurons are green, inhibitory neurons are red, and cells called astrocytes are blue.
That just gives you the degree of this, a 1 millimeter block. There are already millions of cells in that little 1 millimeter block. But because it's transparent, you can follow pathways for long. This becomes a lot easier. And that technology is something that actually we had summer students guided by Prachi, who's sitting right there in the lab, attempting to play around. So we had a little young 10th standard student and one who's a first year bachelor student come to the lab, and we said, okay, we'll give you a fluorescing mouse's brain. Let's try to use the technique to make it transparent, just so that you can see if you can render the brain completely transparent. And right before one of them left today, so excited, because the idea she could take the brain and she could render it transparent. And that couldn't have been done without the Deseroth lab. He's revolutionized what is available for all of us to look at, and it's a matter of time before he gets a Nobel Prize. It's just a matter of time, because it's inevitable for not just this discovery, but for others. He's an MD PhD at Stanford. But what we can then do is, because we can keep some of these sections living, we can actually watch neurons as they're growing. We can watch where they end up. We can watch how they connect. Okay. So I've just given you one gharana. This gharana started with artists who were not neuroscientists. Would not have happened without microscopists, because without a microscope you can't do anything. Without the chemist you have nothing to stain stuff. So without the textile industry, you would not have had dyes. If you had not had dyes, you would not have had somebody who used the dye on a brain section. You wouldn't have been able to see stuff. Without the doctors, you would have had no access to tissue. Without the anatomists who took the time to actually sit down and look, nobody would have put it all together. But today, it's the geneticists and the engineers who are coming in, and the computer scientists and the machine learning people who will use this and automate it such that you're not limited by human ability. You can take data, scale it such that you can computationally analyze it. You don't have to do it manually anymore. So you will make human beings redundant in the exercise of actually figuring out the connectome map. And that's a good thing, because it will take too many human hours, man and woman hours, to actually figure out how to do this. Much faster for a machine to figure it out. So machine learning coming in a major way to figure out what the connectome looks like. Of course, all the machine learning people are excited by this, because the implications to understanding the connectome become hugely relevant to industry. Okay. Now I will move to another gharana. That's a gharana, by the way, the gravedigger gharana has exploded. People are heavily investing majorly in figuring out maps in them. But this is what I call the map maker, truly the map maker gharana, or the hole in the brain gharana. There's a reason why I've called it the hole in the brain. So many years ago, there were people like Franz Gall and others who would have sat in this room and touched your skull and felt a bony protrusion and said, you're very emotional. I would have felt a bony protrusion and said, you are highly intuitive, because there was a profession called phrenology. Phrenology believed in identifying personal characteristics based on bumps in your skull. Okay, now the idea was very wrong, but it was extremely popular, and phrenologists all over Europe, really made a business of figuring out what bumps in the skull told you about what that person's personality traits or characteristics were. So you can see, for example, secretiveness is right over and destructiveness is right over your ear. I don't know why, but they obviously put functions in different places. They got this wrong in a major way, but they got something right. One little thing they got right was that function is localized. Not localized like this, and certainly by touching your skull you can't figure out anything. Okay, all you will figure out is how your skull bones have fused. You will not know anything about your inherent characteristics, but function is localized. That is true, and that matters because every single person in this room is processing vision back here in their occipital lobe. Every single person here is processing hearing, and if you're paying attention to me, it's your temporal lobe. I'm saying, okay, now I've sat for 20 minutes, it's time to, so different functions, different systems in the brain. That part is correct, but none of your bumps is going to tell you anything about your function. But for this to really come about, you need to know the story of Phineas Gage. Phineas Gage was a railroad worker in the US, where when the railroads were being laid, he used to use a little major amount of dynamite to blow holes and lay down the tracks. And in the process of doing that, one of those slots of iron backfired and went in through his brain. Okay, so that's, the skull is actually at the Harvard Museum, where you can see it with a hole because it went straight through and through. Now, the only good thing about the brain is it doesn't have pain receptors inside it. So once it's through and it's gone through, you're not in that much pain, even though you have this thing going straight through your head. Because pain is perceived outside, it's perceived by the brain, but inside the brain there are no pain receptors. So you can actually do open brain surgery under local anesthesia, and if you're poking around, you will not get pain from that poking around. Okay. So, but what happened is that Phineas's personality changed. He was a very polite, very, gentlemanly human being, and he became crude, uncaring, impulsive, irrational, antisocial. It was like an on and off. Suddenly you were dealing with a different Phineas Gage after the surgery. And that led to the idea that the region of the brain where he had the damage, the ventromedial region of the frontal lobe, okay, so it's a frontal lobe, regulates the processing of emotion, of top-down control of emotional circuitry, and those were the original ideas that function may be located in specific places. That has proven to be absolutely true. Your frontal cortex, which expanded hugely in evolution, and so we have the largest frontal cortex that there is going, a very large frontal cortex. This in part of our brain expanded and exploded with evolution. So we've been endowed with the most. We should have the best top-down control over our circuitry. We may not, as you have to look around to see that we have not done very well as far as that is concerned, but it does endow us with the ability to shut down what are considered more primitive, responsive pathways. Okay? Location, location, location. So this whole clan of neurobiologists was figuring out what different parts of the brain do. So people figured out Broca's aphasia, Wernicke's aphasia. This is a lesion on the left-hand side where patients with a lesion can understand but cannot speak. So if you're speaking to the person, they understand everything you're saying, but they cannot speak and respond. Wernicke's aphasia, the person says lots of things. So there's language production is not a problem, but what they're producing makes no sense. Okay, it makes absolutely no sense. And based on where the damage is, you can see the Broca's area is in the frontal lobe there. Wernicke's is at the junction between the parietal lobe and the temporal lobe. So those are the regions. People looked at brains of patients after they passed away, and then they figured out what these circuits were. Now, how will you look at what this disease looks like? So if the sentence is the dog needs to go out, so I will take him for a walk. That is a sentence everyone in this room understands. Walk dog. So got understood it, but could not produce that entire sentence well. The architecture of the sentence, syntax, detail, gone, but has understood the concept completely well enough to say walk dog, which is eventually exactly what has to happen, but you've lost all that architecture of the language. What happens to a person with Wernicke's whose language production ability is not affected but can't understand it? Okay, you know that Smoodle Pinkard and that I want to get him around and take care of him like you want before. Doesn't make any sense, but it's a very complex sentence in its architecture. But you will not walk away with that saying the dog has to be walked. You will not walk away with that understanding. We wouldn't have known this without Wilder Penfield's work, in a sense. These maps in the brain, most beautifully elucidated by Wilder Penfield, a doctor at McGill. So what Wilder Penfield did was, in the state-of-the-art surgery at that time, especially in patients who had epilepsy that was not responding to any treatment, the treatments were poor at that time, they used to take out the primary circuit that caused the epilepsy. They used to go in and actually take it out. But before doing that, because it's a local anesthesia circuitry, they had an electrode and they would go in and stimulate and ask the patient what's going on. So when they did that, they realized, they mapped the entire motor map and the entire system, and they poked and they said, oh, my finger's moving. Oops, you just moved. And the hand would move because as you stimulated this area, the other hand will move, the finger will move. Or if you're touching the sensory cortex and you touch me here, I said, oh, you just touched me on the middle of my arm. Oops, you just touched my stomach. So you can see that there is a map. Your entire body is mapped on your brain. And your brain is using certain parts to process information of touch from different parts of your body. Now your brain thinks that your body actually looks like this. And there is a reason why. Because most of your brain is dedicated to getting information from here, from your fingers. This is how you're exploring your world. So your brain thinks that most of your body is actually your fingers and your lips, not your stomach or your shoulders, because your information processing from there is really not that important. Now think about this from the point of view of evolution. When we were monkeys or our ancestors were jumping from tree to tree, this makes sense. You want to use it for the things that you're using to explore the universe. If you're a mouse, you use it for your whiskers, because they are using this to explore, right? They don't have that much light, and most mice will not run around here, but at night, who knows? At night, they might be wandering around and then they will explore using their whiskers. They use the whiskers to explore the environment the way we use our fingers. If you can't see, what will you do? If you're in a dark room and you don't want to bang into something, what do? You put your hands out and you use them and feel them, right? So much the same way, there's a beautiful test for this called the sensory discrimination test, in which you'll realize that when you poke 2 points close to your lip or your finger, you can, this is one point, like 2 pencils touching, and ask somebody, can you feel 2 or can you feel 1? Very easily, even they were very close to each other, on your fingers and your mouth, you can feel it. If you put it on your stomach or you put it on your shoulder, you won't be able to discriminate whether it's 2 or 1 easily, because those areas are not processing detail of sensory information in the same way. We also know that this is not hardwired permanently. It is responsive to what you do. So before training, if you were to look at the monkey brain, it's a squirrel monkey, you can see that that is the part of the brain that is dedicated to the fingers. So digit 1, digit 2, digit 3, digit 4, digit 5, your 5 fingers, right? On the other side of the brain for this hand, this side of the brain for this hand, because everything wires cross, okay? Now, if I sit and simply use 2, 3, 4, 2, 3, 4, what does that look like I'm doing? I could be playing the piano. I could be using the keyboard. 2, 3, 4, 2, 3, 4, and I just repeatedly do it over and over and over again. Then 2, 3, 4 in my brain expands. So that is why musicians who have trained for a long time are training their brain to expand. No nervous system in a particular way and challenge it. No question, it expands and reorganizes. But there are windows in which it organizes very easily. Later and later it gets harder and harder. If you will reorganize your brain dramatically. You may be very, very bad at it because the critical window for learning a new language is closed for pretty much every person in this room. Yeah, pretty much. Because that early window it's very early. So we've lost the window, but you can challenge yourself. You can really challenge yourself to attempt to learn a new language. So V.S. Ramachandran in some ways can be thought of as the inheritor of this particular gharana because he organized the brain so dramatically. One of his books, which is a beautiful book called Phantoms in the Brain, very well worth reading. He describes, can you see how big the face has become over here? On the left hand side, you can see that the face is taking up a large amount of area because the hand is gone. If you amputate the hand, then the face takes over hand area. Which means now when you touch the face, sometimes you feel that you're touching a hand that doesn't exist. Which is why it's called phantoms in the brain. There is no hand. But I am touching the face of a person who had a hand previously and has a reorganization. Now you say, oh, you're scratching my finger, but there is no finger. Okay. So Phantoms in the Brain, great book. Please read it. I will now switch to quickly talking about Brenda Milner because without her, we would not have this particular gharana would not have moved forward the way it has. So, slightly gruesome video, very not a very nice representation, but bear with it.
On September 1st, 1953, William Scoville used a hand crank and a cheap drill saw to bore into a young man's skull, cutting away vital pieces of his brain and sucking them out through a metal tube. But this wasn't a scene from a horror film or a gruesome police report. Dr. Scoville was one of the most renowned neurosurgeons of his time, and the young man was Henry Molaison, the famous patient known as HM, whose case provided amazing insights into how our brains work. As a boy, Henry had cracked his skull in an accident and soon began having seizures, blacking out, and losing control of bodily functions. After enduring years of frequent episodes and even dropping out of high school, the desperate young man had turned to Dr. Scoville, a daredevil known for risky surgeries. Partial lobotomies had been used for decades to treat mental patients, based on the notion that mental functions were strictly localized to corresponding brain areas. Having successfully used them to reduce seizures, a part of the limbic system that was associated with emotion, but whose function was unknown. At first glance, the operation had succeeded. HM's seizures virtually didn't even improved. But there was one problem. His memory was shot. Besides losing most of his memories from the previous decade, HM was unable to form new ones, forgetting what day it was, repeating comments, and even eating multiple meals in a row. When Scoville informed another expert, Wilder Penfield, of the results, he sent a PhD student named Brenda Milner to study HM at his parents' home, where he now spent his days doing odd chores and watching classic movies for the first time, over and over. What she discovered through a series of tests and interviews didn't just contribute greatly to the study of memory, it redefined what memory even meant. This fact that although HM couldn't form new memories, he still retained information long enough from moment to moment to finish a sentence or find the bathroom. When Milner gave him a random number, he managed to remember it for 15 minutes by repeating it to himself constantly. But only 5 minutes later, he forgot the test had even taken place. Neuroscientists had thought of memory as monolithic, all of it essentially the same, and stored throughout the brain. Milner's results were not only the first clue for the now familiar distinction between short-term and long-term memory, but showed that each uses different brain regions. We now know that memory formation involves several steps. After immediate sensory data is temporarily transcribed by neurons in the cortex, it travels to the hippocampus, where special proteins work to strengthen the cortical synaptic connections. If the experience was strong enough, or we recall it periodically in the first few days, the hippocampus then transfers the memory back to the cortex for permanent storage. HM's mind could form the initial impressions, but without a hippocampus to perform this memory consolidation, they eroded like messages scrawled in sand. But this was not the only memory distinction Milner found. In a now famous experiment, she asked HM to trace a third star in the narrow space between the outlines of two concentric ones, while he could only see his paper and pencil through a mirror. Like anyone else performing such an awkward task for the first time, he did horribly. But surprisingly, he improved over repeated trials, even though he had no memory of previous attempts. His unconscious motor centers remembered what the conscious mind had forgotten. What Milner had discovered was that the declarative memory of names, dates, and facts is different from the procedural memory of riding a bicycle or signing your name. And we now know that procedural memory relies more on the basal ganglia and cerebellum, structures that were intact in HM's brain. This distinction between knowing that and knowing how has underpinned all memory research since. HM died at the age of 82 after a mostly peaceful life in a nursing home. Over the years, he had been examined by more than 100 neuroscientists, making his the most studied mind in history. Upon his death, his brain was preserved and scanned before being cut into over 2,000 individual slices and photographed to form a digital map down to the level of individual neurons, all in a live broadcast watched by 400,000 people. Though HM spent most of his life forgetting things, he and his contributions to our understanding of memory will be remembered for generations to come.
This is Henry Gustav Molaison. All of us studied him as HM, the patient HM. Everyone who's done neuroscience knew him only as HM because we didn't know his full name, because his identity was protected till he died. But this individual changed the way we have understood neuroscience and the neuroscience of memory. It is an incredibly unfortunate accident that happened to a young man where they rescued his seizures, but they left him with a life in which he could form no new memories, no new explicit memories. But the fact that he was somebody that the neuroscience community could access changed our understanding of memory formation permanently. It was a big event to know who this person was, because in all textbooks, he was referred to as HM. Only as HM, and nobody knew his full name. When he passed away, he's considered a hero in neuroscience, because without him, we would have had no understanding. Nobody does those surgeries anymore. That scale of surgery stopped after HM because people realized, my goodness, this is what can actually happen as well, right? So the great detail and care is taken even when surgeries are done to find out what function you will compromise. Scoville took out the entire hippocampus. People now do very fine, restricted removal of tissue because they want to save the person from severe seizures if they're not responding to medication. Of course, medication has changed dramatically as well. Who are the people who are now inheriting this gharana? Edward Moser, Maybrit Moster, and John Michael O'Keefe, who got the Nobel Prize on identifying how we process information about space. And they would never have studied. They are graduate students from McGill, which is where Brenda Milner and Wilder Penfield were. There is a history. These gharanas don't just form. You end up being where you are because of people who have studied something for years before you. McGill has seeded a generation of people around the world who've gone on to do memory research. So John O'Keefe was at McGill, and the Mosers went and worked with John O'Keefe when he moved to University College London. So there's a history of how this actually came about. That is the hippocampus in the rat brain. That is the hippocampus in the human brain, just dissected out. It's called the hippocampus. Hippocampus in Greek means seahorse. You can see why it's called the dissected structure looks like a seahorse. That's why hippocampus. There are many such beautiful names within the nervous system. So what I will quickly show you is that how do people do research now at looking at these maps? They're much more complex than what it was then. You can take a rat, you can implant its brain with electrodes, you can watch while it's behaving and record from the neurons as the animal is behaving, and you can identify those parts of the brain that care about specific locations. So if I'm standing here in this room, some cells in my hippocampus, in my CA1 region of my hippocampus will get active because they're identifying this location. I can move to that location and another set of neurons will get active. And as I walk around the room, and if I stay in this room and if I come back to this room over and over again, I will eventually form a stable map of this region in which specific sets of neurons will record location in these places. These are called place cells. They're absolutely important for you to find where you're going. So from here, you will find your way home. But if I tell you to find your way from here to some place you've never been, Chinchpokli, some small street, maybe with a Google Maps you'll be able to do it, but you know, you'll struggle. But if you do it every single day, eventually you'll form a stable map and then nobody will need to tell you anything, you'll just go there automatically. You will find that your maps for places that you've gone to regularly are strong. For places that you've not gone to regularly are not so great, and some of you will be better at it and some of you will be worse at it, depending on how effectively your spatial maps work. Much the same way, rats also have to form maps. They have to remember in YB Chavan Hall, on the right hand side of this, I might find some food lying on the floor if I go there at 2 o'clock at night. Right? So this is a survival essential. Animals need to know the maps of where they're going to find food. How to run away from something if something comes to catch you. I can get electrocuted in this alleyway. There's a lot of water flowing in this particular, pipeline. I better not die. The rats are climbing up and down Mumbai and managing quite well, remarkably well, right? So how does their brain look when they're actually processing this? And so if you record from neurons, there's a rat there. Those sounds you hear are actually neurons firing. And you've just artificially made that sound, but you can actually hear neurons firing. So blue cells are firing in that corner because the blue cell recognizes that corner. They're color coded to give you a sense which neuron is firing. It just gives you a sense of the hundreds of neurons firing simultaneously in the brain. Blue cares about that corner also. Alright? This just gives you a feel of what modern day research is allowing. We can figure out maps. We know the grid cells that allow us to first form a rough map. Then we have the fine-tuned map that comes next. Imagine about food storing birds. Birds which have to store food for when food is going to run out. They can hide 50 to 100 seeds a day. As many as 1,000 locations in which they are hiding them. Then they have to remember where they hid it, because otherwise it's pointless, because you have to go back and retrieve it, right, and eat it. Not only do you have to remember where you left it, you want to remember the sequence in which you left it, because you want to get to the oldest stuff first and the most recent stuff later, because otherwise it will go bad. So you have to not only remember where you hid it, you have to remember the sequence in which you hid it. You have to do it reliably, quality of seeds, so that you preferentially dig up the better ones first and then leave the bad ones for a little later. This is an incredibly complex amount of information birds do. How about migratory birds that cross huge distances across oceans and reliably do it and find their location? Fish, salmon, large migratory species, they are crossing thousands of miles and they're going back to the site that they laid their eggs at or raised their young. Whales that move from up near the Arctic all the way down to the equator. Small distances, and they are creating maps. So animals create maps and we make maps all the time. Most of you will remember what your street felt like when you were growing up. You will remember who was at the corner, what was the shop, what was your neighbors, what did the alleyway look. Incredibly stable maps. The maps that have emotional attachment to them are the strongest maps. Either places that you loved or places that you hated. Both will be very well remembered. The ones that you don't really care about, you're neutral, you tend to forget over a period of time. They don't really matter so much. But emotionally strong place memories are very strongly remembered. And as you train animals, they get better and better. So the red is pre-experiment. You haven't trained the animal to remember where things are. That is inexperienced, the blue, and the green is experienced. The size of the hippocampus gets bigger and bigger after experience. So like I told you, muscle size can change. I told you representation in brain can change based on finger usage if you are playing the violin or playing the piano. Using your brain to remember location in birds can change volume. So it's not that you can't reorganize it. So if you're not adding new neurons, how is this happening? Some parts of the brain add new neurons, the hippocampus does, but most other parts don't. But you can change the number of synapses. You can maintain and strengthen synapses. You can reorganize synapses, and you do this all through life. Otherwise, you can't learn anything. You won't be only stuck with whatever you know when you're born, because you have to keep adding and changing these circuits. So this ability is called plasticity, and we all have it. It does decline with age. The more you use it, the more you're likely to retain it. And the more you challenge yourself, the better you will be at retaining that, right? Now, this is quite dramatic. If you look at the birds in Alaska versus the birds in Colorado. Now, the Colorado birds don't have so much challenge because weather is not so severe. So they don't have only few locations. In the Alaska birds have to hunt long distances. They're very challenged because the environment is quite challenging. If you now ask them how many inspections it takes for them to find it, the guys in Alaska do better. Because they identify and across long distances identify where did I hide my seeds. This matters. What happens to you and me if you give your hippocampus a workout? So, Eleanor Maguire at University College London. Again, there's a relationship. John O'Keefe, who studied these maps in rats, Eleanor Maguire, both at the same university. University College London has a really profound number of people who are doing this kind of work, very elegant work. What her lab and her group showed was they took taxi drivers in London. If you have been to London, you know that those taxi drivers are the most impressive taxi driver of any taxi driver you have ever seen. First of all, London is full of one-ways. Left, right and center there are one-ways, and they have to remember these maps and they have to pass a test. So they train for 2 years and the taxi driver has to pass a test about knowing where, what street, what is a one-way, what is not a one-way. So they have to really test their spatial memory. So the best way she thought of looking at people for spatial memory, no point catching somebody, University College London professor, he probably sits in the back seat of the taxi and goes. He doesn't have to test his hippocampus. But the taxi driver is the best candidate for this. And see, she took taxi drivers from London and imaged their hippocampus over time. And what she found is that the longer you have trained as a taxi driver, the larger your hippocampal volume. Now that's really interesting, because that one thing, of course, the big debate happened saying maybe you need a bigger hippocampus to be a taxi driver, because to even pass that test you need to do better, which is possible. But the cool part of this test is she's following people with time. So she's able to take their baseline and watch them over time as they train, and it turns out volume increases, which means that like the birds who are storing stuff across for long periods of time, their hippocampus is increasing. Even the taxi driver's brain is morphing itself. So map maker Gharana started with physicians, psychologists, electrical engineers. Without the MRI and the imaging and the PET and the resolution, you would not have been able to do this. Writers. And the reason I say writers is because I would again recommend people like Oliver Sacks, who's written things like The Man Who Mistook His Wife for a Hat. Supremely wonderful book about holes in the brain that result in incredible neurological conditions in which the person can't see his wife's face but keeps seeing a hat. So keeps thinking that there's a hat here to pick up. So, you know, it's interesting what your nervous system can do. Please understand that your entire universe is not the universe that exists. It's the universe that your brain creates for you. We don't see anything other than the visible end of the spectrum. We hear only within a certain very limited range. We and our percept and our universe is what has been created by our nervous system. That's all it is. And your nervous system creates the entire magic that we see in the universe. Animals that, for example, see in the UV range see a very different universe than us. And there are birds that see in UV. There are insects that see in UV. We don't see that end of the spectrum, so we don't see the color that they see. We can't hear half the sounds our dogs are hearing. So we can't hear any of those things because it's out of our auditory spectrum range. Okay? There is a lesson to learn from this Gharana. It is use it or lose it. There's no question about it. That is a phrase that is often used in neuroscience, routinely saying use it or lose it. So I'm going to wrap up very quickly with the nasty stuff gharana. Okay, there's a nasty stuff gharana because it's all about poisons. And without poisons, we would have known nothing about the brain. Again, without those poisons. So without knowing bungaro toxin and the toxins that are produced by snakes, we'd have never known about the neuromuscular junction. The reason you paralyze is because this produces a toxin that blocks all the receptors at your neuromuscular junctions, which means you can't move any muscle. Eventually, that means you can't move even your lungs. So it's finished because you blocked all receptors that allow your muscles to move, including the muscles that move your lungs. So that, without that toxin, wouldn't have figured out the architecture of those receptors or any of that, okay? This is a gharana full of empiricists, chemists, pharmacologists, engineers, physicists, doctors, structural biologists, whole bunch of them, and lots of poisonous stuff. So you've heard of this pufferfish, which if you go and eat this delicacy in Japan, you have to sign that if you die after eating your meal, then the restaurant is not responsible, because there is saxitoxin in the pufferfish. That is, blocks neurotransmission again. Of course, you take it out normally, but if your chef doesn't do a very good job, very experienced chef's taking it out, it's considered to be I don't know why anybody would eat this delicacy, because in the process, if it really works, it will finish you off, okay? So I would skip through this very quickly. Snake venom, the largest blockbuster agent for Amgen is up there. Botulinum toxin, produced Clostridium botulinum. That toxin has made Amgen very, very rich. It has made many people in Bollywood expressionless, because once you use botulinum toxin, you paralyze small muscles, which is why all your wrinkles go away. But you also lose the ability to smile, frown, because you can't move these muscles. So you can inject botulinum toxin, Botox. You have lost all expression on their forehead because they froze their forehead solid after putting in, this Botox made Amgen extremely rich. But Botox also taught us about muscular control in the process, because it's also highly valuable to people who have small nerve pain, because you can paralyze locally. People who have diabetic neuropathy, etc., it actually becomes a therapy. But it has probably made Amgen money more because Hollywood and Bollywood could not have survived that many years of their career without this particular agent. But where is this going? These are the inheritors of this particular gharana. Toto Oliveira, one of my colleagues and absolute heroes in my department, K.S. Krishnan. Krishnan decided to map all the snails along the Indian coastline. Toto Oliveira did it in the Philippines. You must have collected those and never even thought about it. How do you think those snails eat? They have to kill something and eat it, na? And they don't move very fast, which means that whatever they kill, it has to die very quickly. Otherwise, if they kill it and it goes swimming away a long distance, then forget it. It is not going to eat for some months, because it doesn't even have eyesight to move around and find it. Which means whatever it kills has to die on the spot and land pretty much on its head. Otherwise, it's not going to work, which means it needs unbelievably powerful toxin. Toxin that is so fast acting that it's boom and dead, right? That generation of reagents is now being purified, and some of the blockbuster painkillers for cancer therapeutics are coming. Now, you will never think that when you originally start. This is why basic research is so important, because people think, go do work on finding painkillers for cancer, but you will never go look here. But actually, this is where the work results in those. So this is why basic and fundamental research is so important, because sometimes you don't know what you're going to find and where you're going to find it. The blockbusters will come from this. But just see this video because you'll understand what I mean. Very innocuous looking animal.
Crawling along the ocean floor is a cone snail. It looks harmless, but appearances can be deceiving. 4 tubes jut out from its shell. The long one at the top is a siphon, which it uses to inhale water and detect prey. The smaller ones on either side are its eye stalks, and in the middle, a deadly proboscis.
Can you see those?
Concealed within the tip is a lethal weapon.
Look at the angle. Once it goes in, it gets stuck.
A neurotoxic mix so complex there is no anti-venom to thwart it. It's armed with at least 100 different toxins.
That's it. And it's instantaneous paralysis.
When some cone snails go hunting, they like to burrow into the sand and wait. When a meal happens by, the cone snail fires its harpoon. The poisons quickly paralyze the fish. Once its prey is helpless, the cone snail retracts it through its proboscis until it engulfs the fish, swallowing it whole. The snail is no longer hungry, and its appearance no longer harmless.
You wouldn't even think about it. I'm sure all of you have walked along the seaside, collected these shells and admired them, and not even thought about how does the creature that lives inside it survive. So Krishnan identified multiple such toxins along the entire coastline of India, and in the process, with work that was done at IISc and TIFR, battery of potential agents that are novel toxins. All these drugs are called conotoxins because they are derived from the cone snail. Conotoxins are the major drugs in the pipeline for intractable pain. The only other approach is opioids, and you know what opioids do and what the risks with opioids are. Those risks are not there with some of these agents. So these are the major drugs in development. But it's absolutely fascinating to think about how a creature like this and studying a creature like this can result in those kind of insights. And I'll skip this part and end with saying that singularly, if there's one thing I want you to walk away with, I want you to walk away with the understanding that experience causes your brain to change. Your brain is not hardwired. What you do with your brain causes the brain to change, both structurally and in its function. So you can keep your brain really active by constantly engaging it in new activities. You can't do the same activity over and over again. Then it doesn't work. Then it becomes habit. Once it becomes habit, your brain does it like an automatic. Then you don't even think about it. You're just doing it like auto. So if you say, I'm going to do Sudoku and that will help my brain, great. But after 3 years of doing Sudoku, then your brain is just doing it automatically. So you need to do something different. The novelty and the new engagement is critical. So new engagement with people, new engagement with tasks that push you out of your comfort zone is the requirement. Comfort zone is the critical part. If you keep doing it because it's comfortable, then it won't work because then it's just habit, right? Eventually. Just do something that makes you feel a bit like a failure. Like, oh my God, I can't do this. Oh my God, I can't do this. But you'll get better at it and then take on the next challenge. And you find that suddenly, remarkably, your brain steps up to the task because that's its job and that's what it's actually ideally trained to do. It'll do it best when you're a baby. It'll do it less well when you're an adult. It doesn't do it so well when you're aged. But at all ages, plasticity is possible. It's most dramatic in the baby brain. Yeah. And I'm going to wrap up simply by saying, what do I study in my lab? I study the circuits in the brain that produce emotion. We're interested in those circuits that regulate anxiety, depression. Why are some people particularly prone to stress and some people can just buffer stress beautifully? What are the circuits that allow stress to be processed in the brain? How does the experiences you have in life shape these circuits? And we find that the experiences in early life, in childhood and adolescence, have the most profound effect on the brain. The first decade of a human being's life is perhaps the most important in shaping the circuitry for the rest of that individual's lifespan. So that first decade is key. If India did one thing right, which is to look after its babies, its young, everything else gets sorted out. Everything else, now building the roads will happen automatically if you have a mass of people who can do it. If you invest in that, a lot will automatically come. And there's no question that India has the youth of the world. We have the largest challenge in this country because we have the youth of the world. We also don't have the capacity to educate this entire youth and look after them well. Nutrition, these are the kind of things that will keep people adaptable. So we got to switch from performance-oriented examinations to life skill-associated learning. I mean, how do you convert it into life skills so that individuals learn the life skills to survive through life? That approach really needs radical thinking in India. And how do drugs alter emotional circuitry? So if I was to say some neuroscientists are psychologists, anthropologists or behaviorists, or some of them are even storytellers, then this is the gharana that I belong to. This is the gharana that I'm fascinated by, and my heroes are from this particular gharana. And I will wrap up with a very favorite statement from Walter Cannon, who's also from my gharana, which says, "Only when we know what has been done by earlier contributors can we judge the present scene." So this is the history of any profession, provides you deep insights into where the profession is going, because people before you have asked these questions, people who will come after you will continue to ask these questions. And in a single sentence, if I was to tell people why I love doing what I do, it is because I feel part of a tradition of people who have been asking questions for hundreds of years. And I know I'm part of an amazing tradition of people who will continue to ask these questions. So immortality comes not in one's individual self, but comes in the fact that as humanity, you have been pursuing certain questions that are bigger than the individual. And if that doesn't get people buzzed and young people excited, anybody can be a neuroscientist at any age. So there's much that the nervous system and the mysteries of the nervous system offer. And I will wrap up and take questions. Thank you very much.
Maybe 5 questions.
That only the sweet part of whatever you eat, the food that goes to the brain, and the rest of it all goes into the organs and
so 20% of the glucose that utilization happens by your brain. So certainly a lot of it is glucose, but a lot of your fruit eventually gets converted to glucose in some way, shape or form. The other thing that the brain chews up is lipids. So omega fatty acids, polyunsaturated fatty acids, again, they are food for the brain, because just think about the membrane that you would have if one cell is a meter long. The amount of lipid that you need to cover that long distance is not small. So
where do you find
almonds, walnuts. But yeah, I mean, a lot of omega fatty acids. So fatty acids of a high quality and caliber, not the kind that you get in fried food, but the kind that are naturally present are brain food for sure. But glucose
found in bananas, honey?
Not the PUFAs, not so much. So they're there, but you get a whole bunch of other things. A lot of your essential amino acids are in those substances. So banana is full of tryptophan, for example, which is your essential amino acid to produce serotonin in your brain. So yeah, banana will also give you the straight out glucose that you need for your brain utilization. Lots of foods which are your essential amino acids that produce the neurotransmitters in your brain are produced from food. You can't produce those without eating those foods. So tryptophan, tyrosine, these are essential amino acids that need to come from your nutrition. If you're nutritionally deprived, you will end up with not being able to produce those neurotransmitters at normal levels.
But then how do you explain the yogis who go on fasts and, there are those breathing practices and things like that?
So I'm not a big believer in starvation to death, right? Starvation to death has problems. Starvation period for short periods of time and fasting is remarkably good. We were never evolved as a species to eat as much as we eat. We evolved to find food. We were a hunter-gatherer society, which meant that we had to hunt, means you had to walk and find and kill and bring, or you had to go and pluck a berry or pluck a nut or whatever. That means you have to actually move. And because there's scarcity around you, you're not going to be eating every meal, 3 or 4 meals a day for a week. So you sometimes stashed up and then you fasted. So your system evolved to fast. And so there's actually a lot of value in caloric restriction or reducing the amount you eat or even the number of hours you can just fast. But there's a problem with starvation where you go off essential amino acids and essential nutrients for long periods of time. And all the data with starving is that you will cause catastrophe on your brain. But all the data with intermittent fasting and caloric restriction is that you improve brain function dramatically. There is starvation. And unfortunately, sometimes you go the direction of starvation. That's not required. But there's a lot of value to intermittent fasting. Incredible effects on aging. So if you look at, you look at yeast, you look at fly, you put them on a caloric restricted diet, you dramatically increase lifespan. No question about it. Every species that has been tested on caloric restriction increases lifespan. No question about it. So there is a value to intermittent fasting for sure. Yeah.
Autistic kids basically have seen that they're good in one thing and they're trained in that aspect. They tend to do well in that. So what is the thing like?
Yeah. So children with autism, the spectrum, it's an autism spectrum disorder. So you can have kids at multiple levels on that spectrum. You can have the mildest versions like Asperger's, which is probably the most socially easy to integrate into our social structure. Then you can have autistic savants, who are incredibly talented in specific domains of, it could be math, it could be music, it could be certain memory tasks at which you get a remarkable ability, but it comes sometimes at the cost of the social integration. We are a very social species. And the biggest cost and the hardest thing for children with autism is that they don't socially process the universe the way we socially process our universe. And if you notice, we focus on the eyes, the nose, the lips. This is our structure we focus on, and we use this to gauge a social situation. An autistic child cannot gauge environments because their ability to socially grasp a situation is really, really challenged. And it's an emerging epidemic, and it's reaching epidemic proportions, and we don't understand. The neuroscience community truly doesn't understand. Could it simply be that we've missed so many diagnoses, or there is really an emerging epidemic? There appears to be an emerging epidemic in that it's much larger than it used to be. Some of it is also that we are diagnosing a wider and wider. In the past, we might not have diagnosed people at a certain end of the spectrum and would have just said that that's a variation of existent behavior. Would still be called completely in the normal spectrum of behavior. But yeah, but all of those are based on genetic models of autism, which is only a fraction, small fraction of autism spectrum. That is well studied and that is relatively well understood. But all autism spectrum disorder doesn't fall into that category. And so that's where the field is currently. The major area of interest for lots of groups around the world.
Yeah. I want to know like the serotonin and all that is released, and you said that we can even train our brain in certain ways of improving the function. So can we also train it to release these serotonin and other things?
So I would say maybe not in terms of specifically regulating serotonin, but you can certainly train your brain. No question about it. So for example, let's say that, we have rhythms in the brain. There are certain rhythms that are associated with states. So they'll have these certain kind of rhythm activity. So if you look at an EEG, you will have a certain rhythm activity associated with sleeping, arousal. Attention, for example, increases gamma rhythms dramatically in the brain. You can take individuals who've done transcendental meditation practice and you can have them strapped up to an EEG setup. So you are recording. They find it a lot easier to switch straight into gamma rhythms, which is the kind of activity that you see when you are attending to a task. So if you and I are focused on a task, we'll have gamma activity within certain regions of the brain. That form of activity has been shown to kick in much faster in long-term practitioners of TM. Now that's interesting because that means that, and I would be surprised if you can't train the brain to do certain things. I mean, that's the job of that machine. So you can probably train it to do certain tasks, no questions about it. And by doing that, you can move into a brain state which is different. Much the same way you can train your brain to be more resilient to trauma and stress. No question about it again, because cognitive behavior therapy works wonders on individuals who are struggling to cope with stress. And if it can work wonders, it's because you're training your brain to handle environment. And you can't control the environment. You can control what you do with your response to it. So people do train individuals to handle situations around them, and I would be surprised if it did work. You can train mice and rats, so there's no reason why you can't train us to also do a spectrum of tasks. I don't know if you can train yourself into releasing serotonin. No one's asked that question, so I don't know. But it would be lovely if you could. You can certainly take more tryptophan containing foods to make sure that you at least don't get depleted on your serotonin. That you can definitely do.
Where does the yoga and yoga practices come in the picture as far as brain functioning is concerned?
Right. So again, I would say in the spectrum of yoga, which is not just the physical practice of yoga, but the ability to use breath control and to hold specific yogic postures for long durations of time, once again that moves into the zone of being able to train your mind to hold a specific state. It would fall into the category of what transcendental meditation would do. I would be surprised if yogis who've been training for long durations will not also be able to quickly move into a gamma rhythm-like activity. The other thing that happens with yoga, which is perhaps even more dramatic, is you do breath control. And you get a condition probably in the brain. Now I'm guessing, I'm imagining this, I don't know, nobody's ever really tested this. There's this condition in the brain called ischemic preconditioning. So let's say that I have less amount of oxygen in my brain, right? Now, if my cells see less oxygen, very bad situation would be stroke. You don't see any oxygen, cells die, right? But if you have seen slightly less oxygen every now and then your cells get preconditioned. It's like cardiac conditioning when you run. So let's say you've been running and you've been training your heart gets trained to handle lower amounts of oxygen for shorter periods of time. People who do deep water swimming and can hold their breath underneath water for long durations of time are preconditioning their brain to handle short durations of less oxygen. Now there's a lot of molecular and cellular data that indicates that if you train your cells to handle slightly lower amounts of and mildly anoxic state for short periods of time, those cells can handle the second oxidative trauma very well. So it's almost as though you preconditioned or prepped them. And that's true for the cardiac system. It appears to be true for neurons also. So I've always wondered if the breath control or the pranayam practices or the breathing practices are not a little bit like training your brain to handle holding both a state of attention but also transient anoxia. Slightly lowered amount of oxygen for short durations, which acts as almost a booster to get those cells to be toughened in case oxidative stress comes later. Now, there's no evidence for this. There's a lot of evidence in animals where if you do ischemic preconditioning, then if a heart attack happens, you can handle the heart attack much better than if you had no preconditioning. So I've wondered if, there's a lot of oxygen sensing, pH sensing neurons in our brain that detect changes in breath and the level of acid in our blood, which is a measure in a sense of anoxia to some extent. Whether that also regulates the brain, we don't know. Nobody's done that kind of research, but I'd be surprised if some of that effect is not. There's a lot of value in some of those trainings, and I would say even if you don't know what it does, if you experience it, that's good enough, right? Some of it is just, if you've experienced it and it has worked for you, you don't need to know how it works. Half of medicine is like that. You don't know how it works, but if it works for you, take it. How does it matter? You may not know the mechanism. You don't need to know the mechanism for it to work. If you experience it and it experientially proves to be true, I would say there's no harm. Yeah.
Most
of the yogic practices, they ask you to look here from inside. I mean, looking from inside is something that we are always thinking something from outside, and so they ask you, so by doing that, are you stopping some activity or are you activating some?
Don't know, but I would say that you're concentrating. In the process, the degree of the thoughtless state is one of the hardest states to achieve continuously for a period of time, and that requires remarkable concentration. So when you're attending, you get closest to it. Let's say you're focused on one thing, then all other extraneous information goes away, right? That's absolute focus. In some sense, that's asking you to, without using some external tool, to internally find a tool that you can train your mind to just say, I will not have any stray thoughts. That's bringing yourself to perhaps the highest level of gamma activity. That's the kind of activity your brain produces when you're singularly focused on a task.
Yeah, but they always say this spot. So is it
I don't know if that, no, not right here, no. But I think it doesn't matter if it's this spot or that spot. I can take away stray thoughts.
Can this be called meditation? Can this be called meditation?
Yeah, of course. Anything that takes you to, I mean, you don't have to
focus on.
So, you know, I consider doing neuroscience a meditative practice. If I'm not distracted by anything else and my mind is utterly focused on something, it is meditative practice. You can call it whatever you focus on can become becoming a mother with your child and utterly focused on that child with no distraction left or right is a meditative practice. It is, after all, the ability to concentrate your entire mind on one focused task with no distraction. Yeah.
Last question. My question is related to that. When we are concentrating on something here, as Yogis, Dr. Lombroso's theory is there. Dr. Lombroso in criminology, he has cited that in the frontal lobe, when the person is very angry, and all criminals have that frontal lobe cavity. Is it related to that? Because it is negative effect?
No, because there's no data to believe that frontal lobe activation is associated with criminal activity. There isn't that. In fact, the genetic associations with criminal activity are itself problematic. And more as we realize in modern neuroscience, we cannot make easy cause and effect. This gene, that disorder. You realize that things are highly multifactorial and it's never as simple as one activity causing a particular behavioral state. In fact, that does not appear to be the case even in simpler systems where you can genetically perturb. Environment has a profound influence. Perhaps the most dramatic data for this is the monoamine oxidase gene, which was thought to be a gene associated with criminality. And in the 80s and 90s, people talked about this gene all the time. And then they realized that you take the gene and based on the kind of environment in which the individual has been raised, the data all falls apart. So it's such a profound influence of environment. It is not your genetic background. It's the combination of genetic background. You can take individuals who have high risk, but when raised in environments that are nurturing, you override. So that's the thing about the nervous system. It overrides a lot of what people used to consider hardwired. It's not hardwired. It's remarkably prone to change. And that's why the simple associations with criminality have fallen apart completely. Just as a field, that entire field which was looking for rather simple associations has just realized that according to me, that's why we have such a problem with our penal system, which is that you use your penal system to take away people but not to restore them back to society, because we can't deal with the problems, which is why we have done that, because the scales of the problems are too large in their magnitude. But the fact of the matter is environment is a profound reverser, and in fact if you fix it early, you may not even have those problems arise later on. So the early tackling seems to be the most key.
Just last week I read the news. One noted criminal from this area, he has stood first in the examination, which was taken in the jail.
Yeah, because frankly, I mean that's where predictive deterministic statements, which biology used to like to make. If you have this gene, then you will get this disease. If you have this genetic predisposition, this is not true. Because very few things have single gene, single cause. Most of them are highly complex and environment plays a profound role in interacting. So deterministic statements are falling away in biology by the minute. There are very few things. I mean, type 1 diabetes, you can make a deterministic statement. Type 2 diabetes, that falls apart already. Multifactorial genetic risk and environment combination. So people, identical twins, one may show up with type 2 diabetes, the other one is just going to buffer it because of the environmental conditions. So you get a profound effect of environment, and that's, anyway, I'm a big believer that the environment can override much. So
let's stop the questions.
Yeah.
One more question. You did not talk about amnesia, which is supposed to be getting.
Yeah. So.
And I have read a book by a doctor which says that it is only coconut oil.
Coconut oil is certainly a good one, as oils go.
Short chain.
Fatty acids, absolutely.
So it can cross the blood-brain barrier.
So you are going to be best friends with the people sitting in the row behind you because they are all big proponents of coconut oil. See, all these Kerala guys who were extremely upset with these, idiot Americans who came and said this coconut oil is horrible. See, the problem with the diet industry, which I wish they learned from the biologists, which is you can't make such everything in moderation is perhaps not bad, right? But this extreme, this is going to kill you, this is going to be so great that it saves you, doesn't seem to work actually. First of all, most of these studies have been done in some small population. More often than not, they've been done in the West. That is a background and a genetic material that is not the same as us. You are doing a cut, copy, paste of a small study to a big population, right? So this becomes problematic. There's no question that there are multiple polyunsaturated fatty acids and short chain fatty acids in coconut that appear to be quite healthy. Yeah, like I said, your brain is full of lipid. So certainly having high quality lipids in your diet is not a bad thing. Also, what has emerged is that everybody used to say fat is this, you'll get cholesterol and finished and you will die. Now everybody's telling you, don't take sugar, sugar will kill you, fat is okay, right? So part of the problem is the fad association, but sugar seems to be far worse than fat. On the spectrum of severity, sugar is 100% much worse than the fat end of the spectrum. As far as whether it will help with, what I call cognitive decline associated with aging. Everybody gets cognitive decline. So part of it is the expectation that cognitive decline will not happen is a problematic expectation. You don't have as much problem that your knees are hurting when you climb up the stairs because you say, oh, my knees are now hurting because they've been used so much, right? Much the same way some cognitive decline is normal. If it didn't happen, then it would be remarkably shocking. But cognitive decline, you will lose certain rapid working memory. You will lose certainly, it's a natural to be expected consequence. Can you reduce it? 100%. But the most effective thing for that is using it and not being so punitive in your making mistakes. Being brave about making mistakes takes away the stress of forgetting. Now, what is the maximum? You forgot. Now, nothing else. And worse than that, nothing has happened. You didn't kill anybody. You forgot, you forgot. It's perfectly okay. We are all forgetting all the time. We just don't remember what we forgot. When you're worried about making a mistake, then you put it under focus and attention. And as you realize that the more you focus on what you forgot, the worse it makes you feel. So I would say that as a system, because we have so much more of an aging population than in the past, people living in the 80s, 90s and 100s, much more. Because if you look at what life expectancy was and what life expectancy is just going to change dramatically. And it's only going to get longer because we've tackling some of the major killers are being tackled. Okay. So that being the case, you're going to have longer lifespans. You've got to adjust to the idea that you have to cognitively expect different things in those decades of life than you did when you were young. That has not happened. We are expecting 90-year-olds to be functioning like 25-year-olds. Is that a fair expectation? I think as society, we could just say this is a 95-year-old brain. This is great for a 95-year-old brain. Because the range of functions will narrow. You will not expect a 95-year-old person to go skydiving. Even in your 50s, you will think 3 times before going skydiving. But maybe in your 20s, you will go skydiving without even worrying about it. So cognitive decline, inevitable. Wisdom increase, also possible. So, we'll value the wisdom increase of experience with the ability to say it's okay. Less ability, greater ability to have watched life come and go and change. I mean, incredible experience that results with decades and decades of being on the planet. So that, I think, is the problem. I think we are expecting people to fall into very narrow expectations of cognitive performance. And when you don't fit that, then people say, oh my God, cognitive decline. Alzheimer's is much more than cognitive decline. That's when you've forgotten who you are, who your wife is, who your child was. That is a scale of cognitive decline in dementia, which is severe. But natural cognitive decline, where you forgot what you talked about yesterday or where something was or whether you even knew, absolutely normal. Expected to occur and should happen because that's the way the brain functions. With age, that is expected to happen.
So is the Alzheimer and all dementia is permanent? I mean, there are particular people who are more prone to it, or is it because of the circumstances or the stresses that you have?
Again, multifactorial. So yeah, very multifactorial. But one of the pieces of data for Alzheimer's is that individuals who have kept themselves cognitively engaged and cognitively active tend to buffer Alzheimer's and cognitive dementia better. Definite. So that data is clear. So if you've kept your nervous system active and you continue to keep your nervous system active, not expecting 50-year-old or 30-year-old performance, but the best performance you can get at that age, when you can keep the nervous system ticking for a long, long time.
Okay, one last question.
Sure.
This intermittent fasting that you said, is it fasting between 2 meals, it should be more, or are you...
So people seem to think that the most effective fasts are 12 to 16-hour fasts. So our body adapted to fast at night. That's how our body adapted. So it or have a really, really early evening meal and don't eat and let yourself go for a long duration without a meal in. But you know, this strongly depends on your own glycemic index, your own, you know, what your glycosylated hemoglobin is. I would never recommend this to a diabetic. God forbid, if they go into hypoglycemic coma, you'll end up with a crisis. So certainly you want to maintain, you know, normal level, because glucose is your brain food. So if you get to the point where you so there is no need to diet when you're old and forget intermittent fasting. You got to that age, it's already, you won the battle. So forget about it, because it's really not a good idea, because glucose is food brain, you know, for your brain, and fat, some amount of fat is highly protective. So this whole idea of being skinny to the point of absurdity is not required, I mean, in some sense. I would say for younger people, well into their 60s, up into their 60s, yes. Caloric restriction, perhaps having smaller quantum of caloric meals, multiple meals, it appears to, the data is very strong on that. In animals, people have done intermittent fasting. People have done caloric restriction where they just reduce the number of calories consumed with each meal. People have done alternate day fasting. So people have done all kinds of experiments, and it's been done in flies, it's been done in rats, it's been done in mice, it's been done in monkeys. There's an ongoing human study where humans are signed on to do strong caloric restriction. But if it's going to make you miserable, this might not be worth it. You know, you'll live a miserable, longer life. It may not be worth it. It's okay. Having a bit of cake never killed anybody. It's alright.
Could you please comment a little bit on Parkinson's? I know many people
I think lecture was very good and it has invoked many questions. However, somewhere we have to stop, and then you can ask question one-to-one. And let's end the lecture over here. Such men like me. So let us say thank you. And I will announce the next lecture, that is on 21st of September. Unfortunately, that is coming in the period of Ganpati, 13th to 23rd. However, it is a feast to you. It is on artificial brain by Mr. Sachin Satpute.
Artificial intelligence.
Artificial intelligence, yes, artificial intelligence. There's where intelligence helps me. On 21st of September at 5 p.m. by Mr. Sachin Satpute of NKCN, Pune. Thank you.